Let us start with lecture 4 of biomolecules, and today we are going to learn about proteins. So the term protein was first coined by the Dutch chemist Gerard John Muller in 1838, and the Greek word proteos means primary holding the first place, or means the primary or holding the first place. The polypeptide chains with a molecular weight of 10,000 Dalton or above are called proteins, and this definition was given by Berzelius. Well, the bonds, as already said in the previous lecture, between the different amino acids is peptide bond. So how do the amino acids join? So the carboxyl group of one amino acid and the amino group of another join together, perform a condensation reaction. So the water gets out and they form a peptide bond between carbon and nitrogen, and this is facilitated by an enzyme known as peptide transferase. So now let us know about the peptide bond. This bond is formed between amino group of, as already said, and the carboxyl group of another, and a hydrogen molecule is removed, and amino acids are the building blocks of proteins. So let us now classify proteins. This classification will be based on composition of the proteins. First, simple proteins in which only amino acids are present. So its examples are albumin and histone. Secondly, conjugated protein. This include proteins plus non-protein parts. For example, nucleoproteins. These have new proteins plus nucleic acids combined together, and its example is chromatin fibers, where histone and DNA are present together in eukaryotic cells or eukaryotic nucleus. And then comes lipoproteins, where protein and lipid are present together, and its example is chylomicron. It is actually formed in digestion when lipid is absorbed. And then there is phosphoprotein, where there is a protein group plus a phosphate group, and its example is casein in milk. Then there are metalloproteins, where protein is present along with metal. For example, hemoglobin, where protein globin is present with heme, which contains iron. And fifth, there are glycoproteins, where protein is present along with carbohydrate. And the types of glycoproteins, these are glycocalyx. These are present outside the cell membrane. These are kind of examples of glycoproteins, and so these are present in this. Protein is present along with oligosaccharides. And secondly, for example, there are mucin in saliva. These are proteins along with mucopolysaccharides. And then there are protein derivatives. So these were the two types, that was protein, simple protein and conjugated protein, and out here it is protein derivatives derived from proteins. So these are intermediates or products of protein of proteins. So its examples are proteases and peptones, and so these are formed on digestion of proteins. And classification of proteins, another classification of proteins, it is based on the structure of proteins. It is not actually classification of proteins, but the classification of the structure of proteins. And under that we will be seeing the different types of proteins based on their structure. So first of all, there is a primary structure of proteins, which is just the sequence in which amino acids are present in it. We generally actually show the peptides from the N-terminal, where there is free N-terminal. We at first tell that amino acid, then the next one, then the next one, and at the end we tell that amino acid where there is a free C group or carboxyl group, and start with where there is a free amino acid group. So this is it. And since there are commonly 20 different amino acids, so if a protein is for example 100 or 200 amino acids long, then we can say that how many different types of proteins can there be due to the different combinations or permutations. So there can be 20 to the power 20 to the power 200 different types of primary structures of that. And then there are secondary structures, which is a structure formed when hydrogen bonds between amino acids of some chains or different chains are formed, or such things. So an example of secondary structure is alpha helix. Generally is right-handed, sometimes left-handed are formed too, but not in all cases. And so it forms a coil-like structure where there are hydrogen bonds in between. So there are, in the previous one, in between primary structure there is only one bond, that is peptide bond. But when we actually consider hydrogen bond, when we consider secondary structure, we actually need to consider hydrogen bond, because hydrogen bond actually helps in forming the secondary structure after the primary structure is formed. And its examples of alpha helix are where the example of proteins where alpha helix structure is seen, or those proteins which are mostly made up of alpha helix, are keratin, which is present in mammals, in nails, hair, horn and claws. And another is collagen protein. It is present in the intercellular matrix, and it is the most common protein in animals. I don't remember in whom, but in animals it is the most common. Outer than that, I don't remember. And another type of secondary structure is beta sheet, and in it also the same, this hydrogen bond is present. There can actually be two types of beta sheets, that is parallel and anti-parallel. Out here it is parallel, since this is actually matching with this one, and if it just turns around this part, then it would be anti-parallel. And out here, example of beta sheets are keratins of reptiles and aves. Aves means birds or reptiles, that's it. And the scales, beak and feather, I mean in them, in these parts, these type of protein is, I mean this type of keratin is found, which are beta pleated. And after that there is fibroin. It is a protein found in the silk fiber of and spider webs. And thirdly there is another structure, which is the, wait a minute, which is the tertiary structure, which is the, oh sorry, which is the tertiary structure. It is the 3D structure which a single polypeptide has formed. Now the thing is, a protein can be made up of different polypeptides. That is, these polypeptides are not connected by peptide bonds. Different, when we call that a protein is made up of 2 or more polypeptides, when the different subunits or the different parts of it are not connected by polypeptide bonds. So if 2 different structures or 2 different parts are not connected by polypeptide bonds, so we call that 2 different polypeptides are forming this protein, or they might be connected covalently through sulfide bonds, or other than that they might not be connected covalently by different other interactions like van der Waals interaction, hydrogen bond, ionic interactions, electrostatic interactions and all that sort of things. Now tertiary level interactions, I mean the tertiary structure, there can be many kinds of bond, that is written outside, there can be many kinds of bond present in a single polypeptide chain. So this is a 3D structure that after the helix is formed. Now see, actually both alpha and beta, alpha helix and beta sheet might be present in a single protein or polypeptide, we can say. And when these polypeptides, another, I mean single polypeptides which are forming a tertiary structure, come together to form a protein, a whole protein, these are known as quaternary structures. Every protein might not have quaternary structures because they might be made up of single polypeptide. So in those proteins it is only up to tertiary structure. So for the making of tertiary structures, these different types of bonds are needed. At first for the primary, since it is actually made up of primary, and after primary can, and secondary can only be formed when primary is already formed, and when secondary is already formed, only then tertiary structure can be formed. So in this requires peptide bond, hydrogen bond, then ionic bond for the formation of the tertiary structure, then disulfide bonds, and then van der Waals forces. And to give an example of the tertiary level structure, we can say of myoglobin, which is a protein present in muscle cells. I mean, not only myoglobin, all the proteins would have a ternary structure, but we are saying of this because in this particular protein it is not only just a protein, but another organic structure is also present. So that is why we are also saying about this. I mean, saying this, or hemoglobin can also be a part, I mean, example of this. Now fourth one is quaternary level structure. There are 2 or more 3D polypeptide chains joined by disulfide bonds. I don't think that always disulfide bond is needed, although I'm not sure, but I don't think that. And so this also includes the same bonds required out there as in tertiary structure. This is the whole structure of the protein when it is a polypeptide. I mean, it is made up of it is a multimer or dimer. I mean, when it is made up of numerous polypeptides. So its example is insulin. It is made up of two chains, that is alpha and beta, and so this is it. And then the alpha chain is 21 amino acid long, and the beta chain is 30 amino acid long. So in total it is 51 amino acids. And the hemoglobin, it is made up of 4 polypeptides, where there are 2 alpha chain and 2 beta chains. So this is the kind of structure of hemoglobin, where this is the globin part and this is the heme part present out here, which contains the iron. And the alpha chain contains 141 amino acids and the beta chain contains 141 amino acids. So let's go into another classification of protein, that is the function based, which is classified on the basis of function. So there can be different types of proteins on the basis of function. Firstly, the structural protein. So its example is collagen, which is an intercellular protein and one of the most abundant protein in animal world. And secondly, there is keratin, which forms exoskeletons. I mean, not exoskeletons, but the external structures like nails, hairs, claws, etc. And secondly, there are storage proteins. An example of this is albumin in egg. And then there are enzymes, namely trypsin, which is a digestive enzyme, then ribosco, which is a photosynthetic enzyme. Most abundant protein in the living world or biosphere. Can you see the most abundant protein in the living world or biosphere? It is a photosynthetic enzyme. And then there are contractile proteins of muscles, which are the myosin and actin, which helps in the relaxation contraction of the muscle cells. And then there is clotting. There are clotting proteins which help in coagulation of blood, namely prothrombins, fibrinogens. Then there are hormones like insulin. Not all hormones are peptides or proteins, but numerous hormones are proteins. But all enzymes are proteins, but not all proteins are enzymes. Enzymes are those which help in performing a reaction. Generally, it enhances or decreases the speed of a reaction. And then there comes the carrier proteins, which help in transportation, generally across the membranes. An example of this is GLUT. GLUT is a glucose transport protein. GLUT4 is present in muscles and livers, and GLUT5 is present in intracellular mucosal cells. Then there are receptor proteins on cell surface. These are sensory in function. They senses smell, taste, touch. They help in etc. And there are transport proteins which transport certain materials from one part to another of the body. An example of this is hemoglobin in blood, in RBCs in humans, and then other examples are myoglobin in muscle cells. And so here it ends the lecture on proteins. Thank you and have a nice day.